Okay, so now we're going to talk about carrying oxygen in blood, how we get oxygen molecules from the alveoli down to the metabolizing cells. So we already talked about Henry's law, which says that gas will enter into a solution in direct proportion to its partial pressure. And the actual amount of that gas that gets into solution will depend on the solubility. So we can look at that for oxygen and carbon dioxide. For oxygen, you can fit 0.003 mils of oxygen into 100 mils of blood for each millimeter of mercury increase in oxygen partial pressure. Quite a bit more of CO2 because CO2 is a more soluble gas. Neither of those solubilities are sufficient to keep your cells alive. So if you're pumping water through your circulatory system, it would carry trivially small amounts of oxygen and carbon dioxide, and you wouldn't live. Luckily, blood, well, not luckily at all, I'm sure it's carefully designed. Blood has features that allow it to carry much more gas than what it could carry in solution alone. The transport of carbon dioxide is fascinating. I encourage you to read about carbon dioxide transport in blood. But today we're just going to focus on oxygen. So oxygen is carried in blood in solution, but only about 1%, a little bit less than 1%. The rest of it is bound to hemoglobin. Hemoglobin, I'm sure you all know about, has a central globin surrounded by 4 heme portions. And the heme portion is where you have the ferrous ion, and that allows loose binding with oxygen. That means the oxygen go on and off, and it binds on and off in proportion to its partial pressure. So the higher the partial pressure of oxygen, the more likely it is that an oxygen molecule will bind to one of these heme portions, and they're all attached to a central protein core. And you also know that they have a higher affinity for carbon monoxide, which can obviously be very dangerous. So this kind of illustrates how that happens. There's the hemoglobin molecule, the central protein globin core, and the heme portions around the outside, 4 of them. So as you increase the partial pressure of oxygen from a low partial pressure to a high partial pressure, then each of these heme portions of the hemoglobin molecule becomes sequentially more likely to bind an oxygen molecule. So at low pressures there's none, higher pressures there's an oxygen there. It might be going on and off and on and off. As you increase the pressure further, then it's more likely to go on to a second one. As you increase it further, more likely to go on to a third. When you get to high partial pressures, sort of up around 90 millimeters of mercury and higher, then you're likely to have oxygen bound to all 4 heme portions. And the blood actually does change color. If you look at blood that is completely desaturated, it can appear quite bluish, maybe not that blue, probably something like that blue. Usually when you see venous blood, it still has probably 75%, like 3 out of the 4 heme portions has an oxygen molecule on it. So the blood that's flowing through your veins right now has 3 oxygen molecules still on it. So you'd have to actually take blood, put it into a sort of chamber where you've got a 0 partial pressure of oxygen, and get the oxygen off the hemoglobin to see what it actually looks like. It's quite dark bluish in color. All right, so to understand how oxygen is carried in blood, you really need to look at this figure called the oxygen hemoglobin dissociation curve. There it is. So here's the partial pressure of oxygen increasing from 0 up to 100. Not much more oxygen gets on after you go above 100, so we don't need to go beyond there. And this shows the oxygen saturation. So that's the percentage of heme molecules that have an oxygen molecule on them. Notice it doesn't go in 4 discrete steps. So it doesn't go from 0 up to 25%, then up to 50%, then up to 75%, then up to 100%. And that's to do with the fact that as you go through these pressure ranges, then each of those heme molecules has a probability, an increased probability that it's going to bind an oxygen. So you end up with a rather smooth curve that looks like this. This curve is very important. Shape of the curve is incredibly useful for a number of reasons. So first of all, you think about what happens when the blood comes by the alveoli. So it's going to see a partial pressure up there of oxygen somewhere around 100. So all of the heme portions on the hemoglobin molecules are going to become bound to oxygen. So we say that it's 100% saturated. Every available space has an oxygen molecule on it. That blood then travels down to the metabolizing tissue. And if you remember, the partial pressure of oxygen down at the tissue is about 40 millimeters of mercury. So now, about 25% of the oxygen molecules come off the hemoglobin. The hemoglobin releases 25% of the oxygen. That oxygen is then available to go into the cell, take part in metabolism. If you were exercising, the partial pressure of oxygen might be lower. So in exercising tissue, the partial pressure of oxygen might be as low as 20, might even be a tiny bit lower than that. What that means is that when the blood flows past exercising tissue, there's quite a large reserve to get more oxygen off the hemoglobin molecule. So much like that single cell organism that's living in a pond, all it needs to do is exercise to bring in more oxygen. And all this tissue needs to do is exercise. That lowers the partial pressure of oxygen, and more oxygen molecules are released from the hemoglobin. So this steep portion of the curve here is very important. That's what allows tissue to regulate how much oxygen is coming off the hemoglobin molecule. Above that point, it's rather flat, or certainly much flatter up here than it is down here. That's important too. What that means is that you can go from a condition where you've got a normal oxygen pressure of 100 all the way down to about 60 before you get much of a decrease in how much oxygen you can stick on the hemoglobin. So if you had lung disease that results in an oxygen pressure of 60 millimeters of mercury, you're still saturating 90% of the hemoglobin molecules with oxygen. So you've got fairly significant lung disease, but you're still sending a lot of oxygen down to the tissue. It also means you can go quite a long way up a mountain before you get to an oxygen pressure that's going to substantially reduce the amount of oxygen you're delivering to the tissue. But after you go beyond that point, you start to get into trouble. So if you go much higher than that point on the mountain, you're going to start to get into trouble because you're sending less oxygen down to the tissue. When you get lung disease that progresses beyond a hypoxia with 60 millimeters of mercury, then you're going to start getting into trouble. That's why 60 millimeters of mercury for oxygen pressure is a threshold for all kinds of things. So you say that people are starting to get into respiratory distress when the oxygen pressure gets below 60. And the reason for that is you're getting to the portion of the oxygen hemoglobin dissociation curve where you start getting into trouble. So here's a couple of important questions. Is oxygen saturation roughly equivalent to PO2? Because when you go onto the wards, patients are going to be wearing a little device on their finger which measures oxygen saturation. So it'll give you a number corresponding to how much of the hemoglobin has an oxygen molecule on it. Some people tend to make the mistake of thinking that number is equivalent to that number. So they see an oxygen saturation of 90 and they say, well, the PO2 of oxygen is 90. Is that true? Another is if a patient has an oxygen saturation of approximately 100%, so you put that device on their finger, it's telling you that their oxygen saturation is about 100%. Do you know that they have enough oxygen? The answer to both of those questions is no. So think about those questions. We'll talk about those further in the large group session. The difference between oxygen saturation and oxygen partial pressure of oxygen. And why might a patient not be getting enough oxygen when you see an oxygen saturation of something like 100%? Think about those. Oxygen transport problems, so there may not enough hemoglobin, and you'll see that when you look at some of the anemia problems in this unit. You may have dysfunctional hemoglobin, where you've got enough hemoglobin, but it's not able to bind the oxygen properly. And again, you'll see those when you look at the anemia patients in this unit. I'm going to just touch on circulation. Obviously, the heart plays a very important role between getting the oxygen that comes into the lungs. Circulation is what then takes that oxygen and delivers it down to the metabolizing cells. So in there you've got systemic circulation. So the left heart needs to maintain a mean arterial pressure of somewhere between 80 to 100 millimeters of mercury. And you've got pulmonary circulation. The right heart needs to maintain a pressure of about 10 to 20 millimeters of mercury. So as long as the left heart and the right heart are doing that, you're going to be fine. And that's all there is to cardiology. Sorry, that's absolutely not true. But you will get another session like this, giving you an overview of the important or sort of the aspects of cardiac physiology that you should be looking at for that part of the unit.